Biostatistics and Medical Informatics Laboratory at IIT Kharagpur has recently developed automated malaria disease detection software. The software will be used to screen only malaria parasites from whole microscopic image. As you can see from the workflow diagram of the software, that blood is first collected from patients and then slide is prepared at pathology lab. Then digital images of the slide are grabbed digitally using by light microscope. These images will be loaded in the developed software. Software will analyze images to screen malaria infected blood cells only. In addition, the software is able to detect Plasmodium falciparum and Plasmodium. Now we are going to demonstrate the complete procedure. First, the image is going to be grabbed by using light microscope from the prepared slide. As you can see, the slide is kept under the microscope and it will be properly focused at appropriate magnification to grab the proper image. The image grabbing software installed in the computer, where the image is being grabbed and will be stored in a particular folder for all the patients. For every patient there will be minimum 10 to 12 image frames, and accordingly for many patients many images we can store perfectly. Here you can see there are some automated corrections, background corrections of the images that can be done to maintain the clarity of the image. In our developed software, we find the patient information module that takes care of all the patient's informations, as well as patients all the images will be loaded and every image will be selected. You can see the slide bar is showing the image processing is going on the selected slide, and as output we are going to get only those infected cells. You can see there are 2 cells which have been obtained from the whole slide. Performance of this software is the sensitivity and specificity are more than approximately 80%. This software can be useful as a telepathology application, especially in the rural areas where no expert pathologist is available to detect the malaria disease.
At IIT Kharagpur, we are researching a potential non-invasive blood glucose measurement technique based on photoacoustic spectroscopy. The developed glucose measurement apparatus consists of an optical excitation source, a laser, which excites the glucose molecules within a tissue sample. Excitation from the laser generates a photoacoustic wave in the sample, which is detected by a piezoelectric transducer placed in contact with the sample. The piezoelectric converts the acoustic wave generated in the sample to an electrical signal. The electrical signal is feeble in nature and is amplified using a low noise amplifier. Following this, the signal is sampled and digitized using an analog to digital converter. Multiple frames of the same signal are acquired and averaged to reduce noise. This processing of the digital signal is performed on a FPGA platform. Finally, the noise-free signal is transferred to a computer or smartphone, where its characteristic features are measured and calibrated to obtain a final glucose concentration value. The device was initially tested in vitro on glucose solutions at different concentrations. The photoacoustic measurements were found to increase with glucose concentration as shown. Following in vitro measurements, in vivo measurements were performed on a set of individuals suffering from diabetes. 2 blood glucose measurements were taken. one fasting measurement and another postprandial. The photoacoustic measurements again followed the same trend shown by blood glucose concentration of the individuals. Finally, glucose tolerance tests were performed and photoacoustic measurements were performed simultaneously with blood glucose measurements at periodic intervals during the test. The measurements obtained correspond to each other as shown. Calibration of the measurements shows 70% of the measurements to fall within an acceptable accuracy range.